In the case of COVID, the new SARS virus, there are just 3 key publications that need to be studied. By doing so, everybody will be able to see the false assumption that underlies the whole concept of the coronavirus. Furthermore, one will immediately see that the detection method established for SARS-CoV-2 merely detects molecules that are produced in every metabolism.
Nur Dinge nachweist, die in jedem Stoffwechsel produziert werden.
These are the 3 seminal publications on SARS-CoV-2. In 2 of these, the existence of the new coronavirus was allegedly proven, and in the other, a specific laboratory test for its detection in the human body was claimed to have been developed. Virology has long established distinct, logical and verifiable criteria for detecting a pathogen. And of course, science in general also provides clear, logical rules as to when a paper may be classified as scientific. Let's briefly go through these required criteria step by step and review how many of them were fulfilled in the 3 papers on SARS-CoV-2. First, do the 3 publications fulfill the basic formal requirements for a scientific paper? Yes, all 3 of them fulfill the requirements. Second, were the papers peer-reviewed by independent experts before publication? No, this only happened for 2 of the papers. One paper, in which the German virologist Christian Drosten was involved, was published without peer review. Third, were there thorough investigations of other possible causes of the respiratory diseases? No, other possible non-viral causes were not taken into account. Fourth, was a pathogen isolated? No, indeed this would have been one of the most important steps in proving its existence. One paper claims isolation, but nothing was ever done that could remotely be considered as the isolation of a pathogen. Fifth, was a pathogen biochemically characterized? No, how could it be without prior isolation? Sixth, was it possible to determine a pathogen as the cause of the respiratory diseases? No, without prior isolation, this is also impossible. Seventh, is there evidence for the transmissibility of the disease? No. Transmissibility is based only on superficial appearance and unreliable laboratory tests. Eighth, is there evidence that COVID-19 is a distinct, specific disease? No. COVID-19 is nothing more than a mixture of long-known symptoms and entire clinical pictures that can have a wide variety of causes. Ninth, has the claimed viral genome ever been detected in nature, in humans, animals or plants? No. As Dr. Lanka has already mentioned, the genome of SARS-CoV-2 is nothing more than a computer-generated theoretical construct consisting of endogenous components and fictitious segments that have been generated by means of software programs. 10. Is there a validated, reliable, specific detection method for the suspected pathogen that is suitable for diagnostic purposes? No. The polymerase chain reaction, or PCR for short, is a molecular biology method that has been completely misused for the diagnosis of viruses for years. And 11. Was every procedure, every method, every laboratory result and every conclusion substantiated with valid control experiments and control groups? No, there was not a single proper control in all 3 papers. This is the foundation of the so-called 2020 corona pandemic, scientific papers that fail just about every criterion required. No isolation means no evidence of a virus. No control experiments means it's not scientific.